How is it that we find ourselves surrounded by such complexity, such elegance?
The genes of you and me, the genes of you and me, are all made of DNA. We're all made of the same chemical DNA. We're all made of DNA.
Hello, you're listening to DNA Today, a genetics podcast and radio show. I'm your host, Dineen. I'm also a certified genetic counselor practicing in the prenatal space. On this show, we explore genetics' impact on our health through conversations with leaders in genetics. These are experts like fellow genetic counselors, researchers, doctors, and patient advocates. In this episode, we are exploring how to utilize sequencing to assess inherited cancer risk. Joining me for this discussion is Dr. Sandra Balladares. She is currently the HLA Typing Franchise Leader at Thermo Fisher Scientific, and she formerly held positions at Illumina, most recently as Staff Segment Marketing Manager in Oncology. Thank you so much for joining us on the show here.
Thank you very much, Kira. It's a great pleasure to have the opportunity to share my story.
Yeah, and let's start with that. I'd love to hear about your personal experience with breast cancer and just sharing with our audience of that patient angle that you're also coming from.
Yes, sure. So, everything started in May 10, 2010. Obviously, nobody expects to be diagnosed with cancer, but in my case, being a scientist and with a family history of breast cancer, for me, it was less expected, no? And so I was getting ready on the morning of May 10 to go for breakfast. It was Mother's Day on May 10 in 2010. When I was getting a shower and suddenly I felt this lump on my left breast, I got kind of worried. And as soon as I got to breakfast, I asked my sister, who is an OBGYN, to go with me to the restroom. I couldn't really wait to have her exploring me. And after she explored me, she told me, after breakfast, you have to come with me to my office. So I was extremely nervous, no? I was the mom of two babies. They were 1 and 3 years old. And during that time, I was working as sales manager for Life Technologies. And it was a very interesting point of time because precision medicine, personalized medicine was kind of emerging. And during that time, kind of the gold standard for sequencing was Sanger sequencing. Next generation sequencing was in its infancy. It was kind of being developed, but not yet adopted for clinical testing. So that is important as part of my story that is related to genetic testing. So I was well diagnosed, and for important things that are critical for patient survival are that, you want to be diagnosed in early stage. You want to, get the right treatment for your type of cancer. And third, you want to make sure that you survive the treatment, because the treatment is also extremely toxic. And number 4, you want to make sure that if there is any recurrence, that you are aware about the potential chances of recurrence in your case. And genetic testing will play an important role. So, first of all, with the first one, I think that I was lucky because I was diagnosed with a stage 1A type of cancer. So the higher the stage means that it is more spread in your body. So in my case, it was a very small lump. Regardless, the size of the lump, I required a mastectomy and also cycles of chemotherapy, 4 cycles of epirubicin, plus 4 cycles of Taxotere, plus 12 cycles of oh my goodness, I forgot my treatment. Herceptin, because one of the actually biomarkers was HER2 positive.
Unfortunately, there's an underrepresentation of people of non-European descent in genetic studies. We've explored this in multiple episodes of DNA Today. Illumina is also very aware of this issue and how polygenic risk scores developed for people who are of European descent cannot be applied to other ancestries without further validations. To directly address this, Allelica developed new models to improve predictive power in non-European people. Allelica validated their polygenic risk scores in non-European populations to provide ancestry-specific genetic risk assessment. Interested in learning more? Visit allelica.com. Allelica, empowering the next generation of clinical genomics. Do you like science-themed apparel? If you follow us on Instagram at DNA Radio, you've seen how much I love genetic shirts, jewelry, masks, you name it. That's why I want to tell you about Thermo Fisher's Aspire program. It's a rewards program created with scientists like you in mind. Points earned every time you use or purchase lab products. Then you can redeem those points for fun, science-themed apparel, career and educational items, and more. One of my friends in the program got a DNA hoodie, and it was clearly designed by a genetics nerd because you zip up the base pairs. Here's a bonus. As a Thermo Fisher Aspire member, you will receive a free full-size trial product every year. You can also use points to get full-size products from their catalog that you haven't tried before. With over 100,000 kits, reagents, antibodies, and other products to select from, you'll be sure to find many valuable options for your research. Best of all, you'll get a full-size products, not just samples, so you'll have enough to really experiment with. Anyone can join. You don't need to be a purchaser. So check it out at thermofisher.com/aspire-DNA today to cash in those 500 bonus points. Again, that's thermofisher.com/aspire-DNA today for 500 bonus points. See the show notes for terms and conditions and that link. Do you have patients with primary mitochondrial myopathy? There's a new clinical study looking for people with PMM to enroll. People with primary mitochondrial myopathy can experience muscle weakness or exercise intolerance. This study is assessing the efficacy and safety of a drug to help patients walk further by improving their muscle function. Find out about the study for primary mitochondrial myopathy and how your patients can participate by going to pmmstudy.com. The link is also available in the show notes and at dnapodcast.com. Part of the genetic testing that you did was not right away when you were diagnosed, but it was a couple years later, right, of your first round of genetic testing. What were you offered at that time in terms of genetic testing?
Well, you know what? Actually, that's kind of part of my fundamental fight in my story of breast cancer, because when I was diagnosed, I was 36 years old. According to the National Cancer Guidelines, any patient who is diagnosed before 50 is candidate for genetic testing. I was only 36 years old. I was living in Mexico City, and unfortunately, nobody recommended me any genetic testing when I was diagnosed. It was until, well, again, as I was listing the 4 critical stages for cancer survival. So I got diagnosed, I received the treatment, and then, well, it was until the end when I was kind of wondering what are my chances of recurrence is that I learned about genetic testing, no? And that obviously I needed to know what was my genetic makeup in order to identify like what was the risk for future cancers. So unfortunately, I couldn't really get genetic testing anywhere in Mexico when I was diagnosed because genetic testing didn't exist. There was only kind of some labs that were doing genetic testing for research purposes, but not really for providing clinical guidance to healthcare professionals for risk assessment. So that was kind of unfortunate.
Yeah, and you bring up a good point of, other areas of the world, genetic testing isn't always readily available. So, I practice in the US, so I have more of that perspective, but I think it's important to realize that everywhere is going to be a little bit different, especially we're talking about 10 years ago. So just where genetic testing was 10 years ago is a long time in genetics. But you also got further genetic testing later on. So when did that happen?
So my genetic testing, actually, I got BRCA genes sequenced. I think that it was in 2011, and that was kind of a courtesy of a collaborator. So when I got diagnosed, and given that I was working myself in this biotech company, really pushing for genomics, enjoying genomics, and all the change of paradigm that genomics was bringing into the clinical space. So that was for me an opportunity to make something, to make an impact. And so I decided that, okay, in my personal story, in my personal case, I decided to get a double mastectomy, and I think my doctor also recommended eventually double mastectomy because there were some signs of other lumps in my second breast. But so for me, I was fine. So I was thinking that I was getting rid of all the additional susceptibility for getting a second cancer. But you know what? Again, I think that my position was critical, knowing that I knew as a patient that genetic testing was important and that it could save other patients is having a second recurrence of cancer, breast cancer or ovarian cancer. So I decided to make a change. And I traveled at the end of my chemotherapy. I traveled to Orlando to attend AACR for first time. So my goal was to identify who was doing genetic testing and invite that person to do an awareness campaign about genetic testing. So that person was Dr. Jeffrey Weitzel from City of Hope. During that time, he was the director of the Cancer Genomics Center at City of Hope, and he did a super wonderful work on really helping to create awareness around genetic testing and also creating bridges between different labs in Mexico and his lab in City of Hope in order to enable genetic testing in Latin America. So that was a key. And as part of that collaboration is that I got genetic testing.
Interesting.
And it was really, wonderful at the end, but I didn't get it until kind of 1 year or 2 years after my diagnosis. My genetic testing for BRCA1 and 2 came back negative. But obviously different things, many things have changed since that time. Right now, it's not only BRCA1 and BRCA2, it is many different genes that are associated to breast cancer.
Yeah, you bring a good point that it used to be with breast cancer, we would only test for those two genes, BRCA1 and 2. But now if we're looking at, if patients listening to this and they're saying, well, I think, I do have this family history, maybe similar to yours, of other people in the family having cancers, maybe early ages, similar cancers. How many genes should they be looking at on a panel to say, okay, this is a full panel, like this is a good test that's really comprehensive to do? Is there a ballpark number that you would advise?
I would say that the size of panels and the different providers have different contents of genes. I would say that something that is critically important is that the genes that are included in the panel are at minimum the genes that are recommended by guidelines. So since a few years ago, NCCN is recommending to use now panels instead of versus one single gene. And the reason is that there are different phenotypes of breast cancer. And for those phenotypes of breast cancer, there are different syndromes associated to the development of breast cancer. So there is an overlap between some of the features in the different types of syndromes related to breast cancer. And because of that is that now NCCN guidelines and other guidelines recommend to use multi-gene testing versus one single gene. So, and again, I think that it is important that at minimum, all the genes that are recommended are included in those panels. Oncology, and that expands also to genetic testing for germline susceptibility to increase cancer chances, is changing every day. And there are every day or frequently new genes that are added to those lists, and also the variants that are identified with increasing risk to different types of cancer, not only breast.
Yeah, that's a really good point that people should be checking in to say, well, I had genetic testing years ago, it's probably changed, and maybe there's more genes that should be looked at, especially for like someone in your case where had genetic testing but didn't find there to be a pathogenic variant or a change in the gene that makes the gene not work. And that protection for cancer is lower if that gene is not working. Along with the variants, there can also be variants where we say, well, we see there's a difference in the gene, but we don't know what that means. Does that make the gene not work? Does it mean that, no, it doesn't affect the gene at all and it's just human diversity? How do we handle what, we call these variants of unknown significance, or VUSs? How do we deal with these when it comes up with data from sequencing?
Well, obviously, as you expand the search of potential pathogenic variants beyond like one single gene to multiple genes, you get a lot of background data, background noise that could be potentially related or could be not related. So obviously, in order to confirm any association, it is important to perform additional studies. I will say that perhaps right now we are living these case and control studies like on real time, where all different variants are added into the databases. And there are studies in real time performed to identify if those are indeed pathogenic or not pathogenic. I will say that for variants of uncertain significance, it is important that, number one, healthcare practitioners and labs don't take really any medical advice based on purely having a VUS. And it is important for patients to follow up on those VUS with their healthcare professionals. So usually the labs or the healthcare professionals, as soon as new evidence is added into guidelines, they look into their databases and identify patients who fall into a category where a VUS changed from VUS to pathogenic variant, and they would actively contact the patients. But if not, I think that it's important that patients follow up with their doctors and check if something has changed.
And when we've talked about this genetic testing and sequencing, we're talking about like reading the genes and saying, okay, let's read the entire gene, see if there's any spelling differences there than we expect to see. There's two main ways of sequencing. Can you go over like the Sanger sequencing versus the next generation sequencing?
Yes, of course. And that's why, I was adding that it was a very important moment in 2010 because next generation sequencing was emerging, and really like the technology for reading genes during that time in the clinical space was Sanger sequencing. I will say that the main difference is the amount of sequencing data that you are generating. So, and for that, I want to actually use the example of BRCA1 and 2. So during 2010, when I knew that I was not going to be able to get my sequence, my gene sequenced in Mexico, I was thinking, I work in life technologies. I have access to sequencers. I have access to reagents. I have access. I know I have the protocol. I can do it myself, no? But obviously there was this pressure of time, and obviously I was not really like allowed, or I didn't have the time to do that, because basically in 2010 to do the sequencing of BRCA1 and 2, so the genes are large genes. So in total, so we are talking about 2 genes, but we are talking about is 50 exons that basically to analyze all those exons for the 2 genes. During that time, it was needed to do 80 PCRs and then to sequence those 80 single PCR reactions forward and reverse with Sanger sequencing. So at the end, it was 160 sequencing reactions. And in total, like turnaround time, that will take like from 4 weeks to 8 weeks, depending like what sequencer you had, one single capillary or 96 capillars. Obviously, it would be faster, but it was kind of the time to turnaround time to sequence 2 genes. So, and NGS, as I said, was emerging during that time. NGS was used only like for research projects, but I kind of saw, okay, one day this is going to be done with next generation sequencing. And right now, it's really impressive because you can do not only BRCA1 and 2, but you can add like 50 more genes in order to interrogate for other genes that are associated to cancer syndromes. And you can do all of that in 6 hours, like the library prep. You sequence overnight, and next day you have like all the results, no? So you are sequencing massively a large amount of data of DNA, whether it is multiple samples or multiple genes for fewer samples. So it's really kind of the capacity of doing massively amount of sequencing simultaneously in parallel.
And the turnaround time is just crazy. I mean, you're talking about weeks compared to just like one day overnight to be able to do this testing. I mean, that's just remarkable.
Exactly.
It's just amazing how fast this technology changes. And if we're comparing sequencing to genotyping, how is sequencing superior to genotyping? Because now most testing is doing sequencing. Genotyping isn't happening as much anymore.
That is correct. I will say that, well, if we refer to genotyping as only interrogating specific positions in the genes or in the genome, so obviously it has limited discovery power because you are just limited to interrogate those positions and, well, based on the information that has been published about those positions. Whereas with next generation sequencing, if you are doing sequencing, you are interrogating that position, but you are interrogating every single position in the gene. So, which means that you have the discovery power to identify other variations, other polymorphisms between different individuals. So right now, that's kind of the right approach to do sequencing because, again, so cancer is so variable, is mutating all the time, and also there is a lot of variation in the human population that has not been fully characterized. So it's really important to use an open approach that has a discovery power to interrogate and find new variants.
Yeah, that's a good point. And just going back to like the VUSs and like why those come up more now, because for sequencing, we're not just looking at the hot spots on the gene, we're looking at the entire gene saying, okay, is there any differences in this gene that we should be looking more into? On the flip side with sequencing, is there anything that is a limitation that, we're not able to learn about genes with the approach of sequencing?
Do you know what? That's actually very interesting because next generation sequencing has been evolving a lot. And at the beginning, for me, well, I have been kind of seated in the first row looking at how genetic testing has been varying, from moving from Sanger sequencing to next generation sequencing with only one gene, multiple genes, but also different types of variants. Because also, well, at the beginning, labs were using like two approaches to determine the different types of DNA variants. They were using next generation sequencing to determine SNV, single nucleotide variation, or small insertions and deletions. And the labs were using other technologies based on fragment analysis as MLPA in order to determine copy number of specific regions, large regions of the genes, or even copy number of the whole gene. So at the beginning, kind of all labs were doing two techniques, next generation sequencing for only the small variants and MLPA for determining copy variants. So that has changed also because right now there are newer library prep methods that can actually deliver a very good uniformity of coverage across the different genes versus PCR-based technologies. So that really, let's determine how many correlate number of reads per region to units of a copy number. So which is really great. And also there has been an evolution in the bioinformatics tools that paired with these advances in library prep are now enabling to determine copy number variation also with next generation sequencing, which is really great.
Do you work in a lab? Want to receive rewards when you order supplies? Check out Thermo Fisher Scientific's Aspire program. It's a rewards program created with scientists like you in mind. All members receive a free full-size trial product every year. Points are earned every time you use or purchase products. Rewards include science-themed apparel like a zip-up DNA hoodie. Check it out at thermofisher.com/aspire-DNA today. For a limited time, you can receive 500 bonus points. Again, that's thermofisher.com/aspire-DNA today for 500 bonus points. See the show notes for terms and conditions and that link. Did you know you can get actionable results from polygenic risk scores? Polygenic risk scores allow healthcare providers to more effectively help patients lower their risk of life-threatening common diseases. However, since this is a new application, it's easy for healthcare providers to misuse and misinterpret polygenic risk scores. That's where Allelica is key. Allelica has developed polygenic risk scores that provide risk for conditions based on established guidelines. Their software allows healthcare providers to simply upload genotyped data and receive a clinical-grade polygenic risk score report. Allelica makes it clear that the percentile of a polygenic risk score is useless. Rather, it's the relative or absolute risk that matters. Allelica assesses the absolute risk and provides a lifetime or a 10-year risk depending on the guideline of each disease. This leads to easier interpretation for healthcare providers and better care for patients. Learn more in our upcoming episode with Allelica in January, but if you can't wait to find out more, head over to allelica.com. Allelica, empowering the next generation of clinical genomics. If you have patients with primary mitochondrial myopathy, please let them know about a new clinical study looking for participants. Primary mitochondrial myopathy leads to debilitating symptoms of muscle weakness and fatigue, which means there's an urgent need to find an effective treatment. That's why the study was started. It's a new clinical study to test the efficacy and safety of a drug for people with PMM. During the study, participants will be administered daily medication. The objective of the study is to see if people can walk further and complete other exercise tests better after 24 weeks of treatment. You can connect with a patient advocate in PMM today to learn more about the study, including how your patients can participate. Also, stay tuned for a full episode of DNA Today about mitochondrial disorders, including this study, on February 4th. To learn more about the study, you can visit pmmstudy.com. This link is also available in the show notes and at dnapodcast.com. You can also learn more about other clinical studies for genetic disorders through knowrare.com. There are studies for sickle cell, achondroplasia, cystic fibrosis, and more. Again, that's knowrare.com. And another term that's thrown around with sequencing is like depth of coverage. What does that mean in terms of sequencing? Why is it important to look at depth of coverage when looking at a test or a panel, as we've been talking about?
Basically, when you are doing sequencing, you want to make sure that whatever you are reporting, if there is a variant, that variant is true and is not a background noise. So different labs have established different kind of parameters about what is noise and what is signal. Usually, it is considered that it is needed 100x of coverage in order to call with good accuracy germline variants. But that could also vary from lab to lab and also from some of the regulatory bodies. They could have also different thresholds for detecting variants versus noise.
So it all kind of depends on what that depth of coverage is. There's no real standard yet.
I would say that the kind of the low bar is set like at 100x. However, I know labs that they like to prefer a higher coverage in order to have a higher rate of accuracy in reporting what they consider that is true with a good level of accuracy.
And is there other aspects that labs should be considering when deciding what type of sequencing they're using and what types of panels? Are there other, on the more like lab side that we've been talking about, factors that should be considered when looking at testing and looking at this data?
I think that it's important to consider what is the base call accuracy when it comes to the sequencing platform. When it comes to the content of the panel, well, again, you want to make sure that you have actionable genes recommended by guidelines at minimum. You want to make sure that the test is sensitive, and reproducible. But you also want to make sure that there is a full solution, because analyzing like that vast amount of data is not really something easy. So it is important to have a secondary analysis in the bioinformatics side to analyze all that data, and tertiary analysis in order to interpret like all that data after secondary analysis and to get a biological interpretation of what is that you are getting from the sequencer.
And another topic that I wanted to kind of go back to is when we were talking about your own genetic testing, and that there's germline testing to see if, what you're born with, your genetics, versus somatic testing of, possibly variants, mutations that people have acquired over time. So with cancer, like, okay, what's different about the cancer genetics compared to a person's genetics? So when we're looking at testing for this, usually when people are talking about genetic testing, they're talking about germline, they're talking about anything that's been inherited. You know, what did they get from mom or maternal sample and their paternal or dad sample? Do you see that somatic testing is expanding, that this is happening much more than it used to be? Like, what are the changes that you've seen in terms of like somatic testing being used in healthcare and used to help with like treatment? You said for yourself of looking at like genetic testing, and that helped with one of the treatments that you had for your own breast cancer.
Absolutely, and I think that's kind of one of the paradigms that has changed with the advent of next generation sequencing. So cancer is a disease of the gene, so we are basically accumulating mutations, and so really next generation sequencing is kind of the best technology to identify what are those changes that are happening as a result of the malign growth in the cancer cells. And in that case, yes, we are talking about a second type of genome because it's not the germline genome where we look for association to increased risk to cancer. So in this somatic genome, we are looking at the tumor cell and all those changes that are driving like all the cancer itself.
Yeah, it's interesting to see with
NGS is the right technology to identify all those mutations.
Yeah, I can see this happening much more in the future too, of just like everybody that it's standard, that everybody that has cancer that has somatic testing done and comparing it to germline. Because as you said, you can, for breast cancer, you could have a BRCA mutation, but that's not something you were born with. That's just isolated in the cancer, which I think is something that a lot of people don't know of. You know, they just hear of BRCA and they think, oh, I was either born with it or not, but there's a whole other level to it in terms of the cancer genetics there and everything.
Totally. Something else that I would like to add, and is that unfortunately genetic testing is not really widely adopted yet. There is an opportunity in order to include populations. There are disparities, and not all the populations are really very well studied. So it is unfortunate that there are some gap access, some related to availability of the test. So I can tell you, it's still, 10 years when I was not really recommended genetic testing. And I can tell you that still like now, 11 years after that, I know that there are cases where genetic testing is not recommended in some of the developing countries. So it's really super important to work and enable access to genetic testing in those populations. So I think that, we need a big effort in order to make the test accessible to patients, healthcare providers, still increase awareness around genetic testing because, it is, again, super important and it will save lives and prevent other types of cancer. But it's really critical that there is awareness, and there is still a lot of work to do in that there.
Yeah, I agree. And I think the more healthcare providers that are aware of how important the genetic testing is, as we've talked about today, I think that's going to lead to more people offering this testing, more insurance coverage, more hospitals offering that as, oh, you can get your blood drawn here for a genetic test. And bringing that up so that patients know that not only is it an option, but it's a really important one to be considering and could change treatment and everything. So I think you really highlighted a lot of important aspects with genetic testing and not just, of like, okay, looking at how we're doing this and which is the best, but just like we need to have it in general. So I think that's really, really important to really drive home. Yeah.
Exactly.
And people can learn more about using sequencing to assess inherited cancer risk by visiting Illumina.com. You can learn more about the show by heading over to DNApodcast.com. And you can connect with us on Twitter, Instagram, YouTube, Facebook. The video for this episode, the video interview, is available on our YouTube. And any questions for us, you can send it to info@DNApodcast.com. We'd really appreciate rating and review on Apple. And wanted to thank everybody for listening. Join us next time to learn and discover new advances in the world of genetics.
The genes of you and me, the genes of you and me, they're all made of DNA. We're all made of the same chemical DNA.